we talk today about how we challenge brain cells, how many neurons and glial cells, because there are two types of cells in the brain, through controlled microenvironments. So part of this work has been done in the Institut in Grenoble, and more recently, because I moved very recently to Institut Curie in this laboratory. So, just a first introduction on neurons. Why neurons are quite fascinating cells, at least for me, because first they are very branched cells and long cells. They can be very long. The motor neurons in the human body can be 1 meter long. They are also electron-energetic cells. They can transport electrical signals. And they are somehow the central element of multi-scale field, starting from the brain at the higher scale level and going through one of the lowest levels to the synapse that we heard this morning talking about. So the synapse is mainly the discontinuous structure between the axon and the dendrites, and it's a gap by occurring mostly the transduction of chemical to electrical signal. So we want at some point, we may want to work with in vitro cultures, and one of the most popular systems that we use is our cortical or hippocampal cultures. So the cortex is here, for the mouse embryo, right embryos. And either we take the cortex or we take one of the middle part of this structure, the cortex, and this is the hippocampus neurons. Here, the hippocampus that we heard about, because the hippocampus is where the short-term memories occur, but also is responsible for localization of space, that you probably heard about in this conference. So when we got here the tissue, what can we do? We can dissociate the tissue, put the cell in culture, and this was the sequence of events that occurs in culture. So in the first hour we see laminipodia around the cell, and after a few hours later we have the emergence of neurites, small protrusions here around the cellular body, it's called the soma. After 1 or 2 days in vitro, we have this striking events that is mainly a breaking of symmetry at the cell level. And one of these neurites is growing faster than the others and will become the axons. And then we have the growing of this branch, become the dendrites, and after most 2 weeks in vitro, we have maturation of this dendrite and the formation of synapse. So, and we end up, we do nothing, we just plate the cell at the bottom of Petri dishes, we end up with this structure here, which is a non-structure, like the random localization of cells, and it is nothing to do with very high structures that we can find in the brain. Here, again, the hippocampus, we see that cells are completely aligned here in the cellular body, and the dendrites are also aligned inside the structures. So we would like at some point in vitro to reconstruct this exquisite structure of the brain through controlled microenvironments. So the work that I'm presenting now has been done in Paris by my colleagues, and mainly they use microfluidic chips to confine, to compartmentalize the culture into here 2 compartments, A and B, and to favor the growth of axons from A to B using this type of microstructures. So you can see here that only axons can grow through this very long channel. This channel is about 1 millimeter long, just because axons are longer at this first stage of development than dendrites. So mainly the only protrusions of the cells that can pass through all these long channels are axons. And they worked here to send the axon from A to B, but not from B to A, just by implementing a sort of control access of the axon of the B population by narrowing here the channels here, and I think the channel is quite wide here to collect the maximum axon from these limiting channels. So I can present to show you how it grows in vitro. So here you see the emitting chamber, and you can see this spot here that corresponds to the tip of the axons growing inside the channels. Okay. Oh, of course, these networks have been proved to be functional. This is the work of these 2 PhD students in the group of Paris. So it's using optogenetics and calcium imaging. You can trigger electrical activity inside these two chambers, and you can also image this electrical activity, so calcium imaging. So if you do, for example, excitation of this chamber here in blue, you get this blue signal, and you got also the red signal because of the propagation of the activity in the red chambers, and due to the unidirectional direction of the axons inside the structure. When you excite here the red chamber, you get a signal in the red chamber, and no signal in the blue chamber. Meaning we achieved, in this case, what we wanted from the beginning. But it's not working in all the cases. In about 2/3 of the cases, unfortunately, we also got signals when exciting the red chamber into the blue chamber. This can be suppressed by putting drugs into the system that enhance the bursting level of the structure. But of course, we have to work, and we are currently working on the design of the channel in order to suppress completely the effects, and we have to get at the end a robust system. Okay, so in these studies, the axons were guided because they were long. That was, they were existing in the chambers, and they could go out of the chambers just because of their length. But, sorry, just a small, another study also performed in the group, where now we worked on these channels, but with open space, and with through a combination of chemical and topographical patterning, we can confine axons and cells into these green areas, and we get almost the same types of pattern that we got before, and the neurons here, cellular bodies and axons on this nice tracks here. So yes, the axon was selected here because they were long, but one of the challenges, one of the questions is what can we do if we want to now work at a single cell level, meaning to control the polarization. Here we have in culture a stochastic process of polarization. And I mean that here you have 5 neurons, and one of them at random will become the axons. But of course in the brain it doesn't work like this. There are many signals, chemotaxis, apoptotaxis that select, not at random, one neurite to be the axons. So how can we do in vitro to control this process, and to make that this particular neurite, for example, become the axon? This is one question concerning polarizations. And there is also other questions about branching that is much less explored questions. It's how branch can grow, why do they grow, and why do they grow here and not here, for example. And this is a project that I was very happy before leaving France to know that this project that we proposed was funded, and so we will have a project to create de novo branches from a main shaft here, and to study by super resolutions the dynamic of the cytoskeletal branching point. So I close this is about branching, and I go back to polarization. So how to work in vitro in order to control which neurites, which developing neurite will be the axons. So for that, what we have done in Grenoble was to look at the literature and to see what are the particular characteristics of axons in vitro. And we found this nice work here, showing that plotting the ordinal change of axon directions during the time window, and mainly the distribution of the change of orientation of the axons in time is peaked around 0. So that means that mostly axons go straight. So we decided to use micropatterns of adhesion to constrain the shape of neurons. So here you see in red where we put the adhesive molecules, non-specific adhesion, polylysing of the ornithine, for example. And in white, we have the non-adhesive areas. So here is a controlled pattern. And in this set of patterns here, we change progressively the curvature of 3 of these lines, where neurite will grow, and let one neurite, one stripe being straight. So it's, I will show you how it looks like a cell the neurons growing on this type of pattern here. So you see 3 curved neurites and 1 straight here. And we counted how many axons we found on the straight part here by immunostaining. There are specific markers of the axons, mainly microtubule markers, specific to the axons. And we obtain this graph here. So what we see here is that above a certain curvature, we start to have a significant effect. I mean, we can bias the system only with geometrical constraint and start to have more axons than random along the straight line here. So in these 4 patterns, we reach something like 55% of axons with the straight line. But this was not enough, so we worked more. And we constrained, for a reason that we will not explain here at the time, constrained the soma here into a triangle. And then we obtained this 3-branch pattern, a rate of axons along these directions, reaching something like 90%. So with this, we can imagine constructing in vitro networks, neural networks at a single cell level with this geometry. So here I show an example of neurons, of networks or made of 3 neurons, where the axon is labeled through a specific marker of axonal state. And you see here that the axons are only along the straight directions. Okay, so in the same line of reasoning, we were inspired by an event that occurred in the embryonic brain, that is neural migrations along glial tracts. So mainly this occurs in 1 dimension, and it's quite easy in vitro to try to reproduce this by simply making stripes of adhesions. So we did this varying the width of the stripes from 2 to 8 microns. And this is what we obtained. And what our great surprise, and I'm still surprised by these results, is that neurites accept to spread until something like 8 microns. After that, they undergo some instabilities in their shape, and they tend to go back to their spontaneous diameter in vitro. But they can extend up to 10 times their spontaneous diameter in vitro, which is probably not trivial. Okay, so we see, in the next, you see here, these cells are quite typical of what we observe. I mean, that the larger we make the stripes, it seems that the cells were shorter. And this is what we quantified in 2 situations. We have simple stripes, or when we have stripes with dots, in order to constrain and to confine, to stabilize the cellular body. And we end up with the same type of linear variations, showing that the total neuric length decrease, indeed, with the stripe width. But you can note here that when we decrease by a factor 3 the width of the stripe, we do not decrease by a factor 3 the length of the neuron. That means that the adhesive surface is not conserved in this system. Okay, so what can we do with this knowledge? For our initial question of how to master symmetry breaking during neuronal polarization. So simply what we have done is to break the symmetry of the pattern and to use asymmetric pattern. We use 2 micron thin stripes on one side and 6 micron wide stripe on the other side. So here many data, but basically what this graph shows here that even if all neurons tend to be asymmetric quite soon in culture, because one of the neurites will grow and be an axon and the other will transform into dendrites, the neurons that we plate on this pattern here are the most asymmetric ones. And when we labeled the axons in this system, we found that on many cells, at 100%, the axon is always on the thinner stripes. So with this very simple design, we could here set a priori where the axon is when the cell is still. Okay, so now I want to switch to the mechanism that we guess. Why do we succeed to localize, to inhibit the axonal transformation on curved? Why do we set the axon on the thinnest lines? So we found, in fact, this, when looking to videos, a very striking phenomenon that was called 10 years ago actin weight. It was observed in vitro neurons. And I will show you here a movie on the curved neurites. So you will see at some point a structure coming from the left, yes, here. And when the structure arrives on the curvature, it makes the neurites escape from the initial pattern. And there is a sort of mess here for a while, and then eventually it goes back to its former geometries. So here the same thing, that lower magnification, and what you see here, that when we have this event, you see that the neurites here retract. So each time you have a wave, you have this event coming and a retraction. So the waves here prevent the growth of the neurites on curved structure. This can explain why we have an inhibition of axonal polarization on the curved structures. So what happened now on stripes? So here is a movie. So you can see again these waves propagating along the neurites here. So there is two striking phenomena in this video. One is that the waves, when they travel, they pull on the soma, so they seem to exert force. And the other thing that is not shown on this video is that when we can compute quantitatively that when a wave arrives at the tip, it makes the tip growth. So it seems that the waves are completely associated to the growth of the neurons. So we expect that the more wave we have, the faster the neuron will grow. And so we computed the periodicity of this wave, so mainly the interwave interval in time. So we got the distribution of this interval according to the stripe width here in blue, and we saw that the mean period of these waves increased when we increase the stripe width. So the waves are less frequent when the neurites is larger. So this explains qualitatively what we observe in length, but of course, quantitatively we have to work more on this. Okay, just a few words, because maybe I have 1 minute. Okay, about volume. Now, we talk only about what happened in 2D, but of course, what happened in 3D, when we constrain a neurite to spread on a larger pattern, it's thinner or it's thicker. So we worked in further with a phase image, I mean interferometric microscopy, but with this, it seemed that neurites on the larger stripes are not thinner but wider, bigger, which was quite surprising. But the phase signal gives you information of the age of the cell, but also information on the index, refractive index of the cell. So we cannot say for sure that the neurites here that is larger is also higher. So we are now developing sort of gels for cells, confining cells into microfluidic chips, and filling this chip with fluorescent dyes. And when you take a picture of this, when we record the fluorescent signals of this sample here, the thicker is the cell, the less you get fluorescent signal from the dextron here. So this image completes the volume in real time of the cell. So we can follow the volume of the cell here. So I will just show you a volume movie of a cell. So this was done with the first design that we had, where the ceiling of the cell was quite high, 9 microns. So we don't have, it's not very accurate in volume, but still we see the neurites, and we see, if we work a bit on the signal, we see the actin wave, and now we see the actin waves in volume. So we have to, now we worked on structures that are much less high, and we have better contrast, and we have to work with the cross-section of the neurite here. So you see that we obtain now real aids that are less than 1 micron, and we have to work on the flatness of this chamber in order to have less noise. Okay, so I see my time is over. We will talk about rigidity patterns and glial cells and proliferation. I just want to end with one announcement concerning France and PhD program. We have starting quite soon a PhD program that is funded by the Europe and by the Institute Curie. And this is for interdisciplinarity subject. It's called intersectoral exposure, that company has to be interested, but that to pay money, so it's much easier. And it has to be correlated with international mobility. It means that it is designed for students from abroad France. So this, the first program will launch officially in December. So if you are interested by spending 3 years in Paris for a PhD in biophysics lab, please contact me. And thank you very much for your attention.
Thank you, Catherine. It's open for discussion.
So how does the cell know to make only one axon? And can you force it to make 2 axons by making 2 straight lines?
No, it's very rare event. Sometimes we have 2 axons, but it's less than a few percent. So there is something that inhibits. There's some inhibition. When the axon is formed, it inhibits the formation of the axon.
Does it go through the cell?
Yes. And in fact, observing the waves, we have maybe one mechanism for that, because we observe that during axonal polarization, waves are essentially directed along the axons. So if there is no waves along the right, the right cannot grow. They cannot make another axon. So this can be one answer at the cell level. But of course, there are molecular mechanisms, like diffusion barriers.
So you measure the frequency of waves.
Yes.
Did you also measure the speed of waves? Because these are traveling waves.
Yes, we measure the speed of waves. They are traveling at about 3 microns per minute. And it's
Does it depend on width at the point?
Yeah, it's a bit dependent on width. Not as much as the frequency, but they tend to be a bit slower on larger ones.
Yeah, thanks.
From 3 microns per minute to 2.5 microns per minute.
Okay, so there is no other question. Okay, so thank you, Catherine.